So I realize this is a workshop on cancer, and I am not speaking about cancer. I haven't done no work on cancer, but I was asked by the organizers to give a talk, and I thought that this particular topic might be of interest. This is a problem that I'm working on right now with a chemist at UCLA, and it's on DNA aptamer design. And I'm going to talk about how we've been using ideas from subgraph matching and machine learning to design DNA aptamers. I have to tell you what those are. Okay. So this, and also I wanted to mention that this work is funded by the National Science Foundation, the Simons Foundation, and various NIH organizations. My personal funding, the primary source of funding is the Simons Foundation for this work, but there's also a bit from NSF and these other agencies. The Simons Foundation was really responsible for us starting this collaboration. We couldn't
They
have done
start already
that.
without me? Okay, that's fine. Sorry.
Hi guys, welcome to my
Okay,
talk.
next speaker. Sorry, was out here in the sun.
Okay, very good. All right, so let me go on. So, okay, I got to move my bar here so I can see what my slides say. All right, so what are aptamers? So aptamers are single-stranded nucleotide polymers. They're basically single strand of DNA or RNA. We've been working with DNA primarily. And they're chosen, designed, selected, however you want to call it, for high affinity of binding to specific molecular targets. Also selectivity. So if it binds to everything, it's not really going to help you that much because you don't know what it's binding to. They're typically small in size. They're very reproducible, and importantly, they have a lot of biocompatibility, low immunogenicity, and and a fair amount of structural stability relative to proteins. So it makes them advantageous for biosensing in vivo and for therapeutics. I'm going to talk about a particular method that's used to identify candidates of these for these aptamers. And this is really a very, very combinatorially complex problem because you have no idea what to do at the beginning, right? So typically you start with a library that has a huge number of sequences, say over 1 billion, and then you try to find a way to down select from there, and you may find nothing, right? So because the random sequences may have nothing in them that actually works for what you want to do. So it's a bit of a hit or miss. We're trying to increase the likelihood of having a hit by adding a machine learning component to this. Okay, so the method that we're working with is called SELEX, a systemic evolution of ligands by exponential enrichment. This goes back to the 1990s. The process, as I said before, starts with a combinatorial library with randomized sequences, typically flanked by two constant regions, which are useful to have for this process, and they may also be useful for other aspects of the chemistry. Okay. And yes, and also for next generation sequencing, which has been a big change in the way CellX is done since about 2022. So having next generation sequencing has made it's kind of catapulted CellX back into the forefront because it greatly has the chance of increasing its success. Even so, with the current methods without machine learning, CellX is successful roughly 30% of the time. So let me describe how this works. Typically, you have a process where you start with this big library, and then you have so you have this big library they're all sort of living in a soup. You have a substrate with your target, and then you're going to see what sticks to the target. Then you're going to do some kind of PCR amplification, and then you're going to then start with a new soup and just iterate on that many times, like 20 times. Um, and at various stages, you might do next generation sequencing, but in particular, at the end of the process, you'll do next generation sequencing. And the next generation sequencing is going to output a list of aptamers that's far less than the billion that you started with. It's going to be maybe several thousand. We have an example with 500,000. And you get this list of candidates, and along with the list, you get a number count. How many times does a particular sequence show up after this 20 iteration process? So in sort of the classical method, you would take the ones that show up the most, maybe a couple of them, and then you would run the individual experiments for affinity and specificity. So we're injecting a step in the intermediate step here, which is a machine learning step. And in particular, we're trying to test some hypotheses, one of which seems to be testing very well. The hypothesis is that the high number count is not necessarily the most, like, it's not necessarily the only indicator that this might be an interesting aptamer for the particular target we're interested in. So, for example, we're hypothesizing that there may be aptamers in this list here that may only have a count of one, but yet they turn out to be excellent choices. So, how are we going to find those? So, we're going to find those by doing some very clever machine learning. And so, in order to do that, we also need to be able to identify folding structures for these DNA, single-stranded DNA. So, let's talk about that. So, first of all, I want to talk a little, just mention a few words about why these things fold. So DNA is like sticky tape in the sense that it's formed from it's basically a sequence of nucleotides, and there's only four that you can have on your strand, and each of the four have a are they come in pairs. They really like to stick to their conjugate pair. And so that means that along this strand of DNA, you have many copies of nucleotides that want to stick to another nucleotide, which means that, if it gets a chance to fold, it might just fold very nicely and be stuck together. Okay, so and that folding is basically the secondary structure. So we'd like to be able to get all the secondary structures very quickly and use that information to then do some machine learning. So when we started working on this, the state of the art at the time was called mFold, which is a proprietary algorithm for computing secondary structure that has a internet GUI interface. You have to literally type in by hand the sequence that you want, and then it comes back with a candidate secondary structure. So obviously this is going to be, not so useful when you need to get 5,000 of them all at once. Of course, you could try to get that code and, go under the hood and engineer it, but we decided to try our hand at writing a new code for doing this that we would put in the open source. So this is now available. It's called GMFold. It's a Python code, and it's available on a public GitHub repository. Okay, so let me talk a little bit about how that problem is solved. So the DNA, so it turns, and we actually used a little bit of ideas from modern inexact subgraph matching optimization, which is something that I've worked on outside of this space. I had a DARPA program that was related to adversarial activity detection, so it was very different from DNA. But I learned a lot about subgraph matching and the best performing algorithms. So that helped us to speed up a little bit these algorithms. So basically, DNA secondary structure is a bipartite graph matching problem. The graph is the DNA strand, which is basically a sequence of the nucleotides. So it's a linear annotated graph in which each node is labeled with the DNA nucleotide. The edges are the DNA backbone of the primary sequences, and the nucleotides have conjugate pairs, A and T and C and G. And so you're trying to figure out which of the A's are going to bind to which of the T's and which of the C's to which of the G's. And the goal is to figure out how to do this so that the binding that you choose has the lowest free energy structure. And the free energy structure information goes back to the 1980s. Um, okay, I don't think I'm going to need to say too much about subgraph matching methods, but the ones that I've worked on are the constraint propagation methods, which are typically the fastest for larger graphs, meaning like like along the size that larger would be, I mean like 50 nodes. It's not very large. Um, that's for the subgraph in a larger graph. Here we're doing bipartite subgraph to subgraph, so it's a little different from some of these problems, but nevertheless we can use some of the constraint propagation ideas. So here's an example of using a constraint. It's very simple. So this is a serotonin aptamer, and it's a little. I've got a lot of little tiny letters here, but there's no other way to do it. So if you want to do the matrix, so what we have is the sequence here and the sequence here. It's the same sequence, and so we're trying to come up with a matrix that shows which of these nucleotides bind to which other ones. So the first thing you can do is you can start with this blank slate and you can color in all the boxes that would connect the conjugate base pairs to themselves. So you do that and you get this, right? And then what you do is you actually dig into the literature on aptamers and you discover that in terms of the low energy states, they typically try to form these necks in little pieces or big pieces, but it's very common. In fact, it's rare to see two nucleotides binding to each other without another pair next to them. So almost always they come not only in pair to one to another one, but another pair right next to them. So if you literally demand that as another constraint on the problem, suddenly this bigger matrix becomes this, it becomes much sparser, right? So just doing that step alone can actually speed up your algorithm by a fair amount. The other thing that you need to do is like if you get under the hood, you need to look at the fact that the energy calculation is based on phase classes. So these are just shapes for the two-dimensional structure, like a hairpin loop, an inner loop, a bulge, or a multi-branch. Um, and then you basically have a recursive process for minimizing the free energy, but we're already starting at a better place in this process than some of the older algorithms because we're limiting our search to these possible combinations. And I'm not, because of the time constraint, I'm not going to get too far into the weeds on the iterative process. I will say this, even though it seems like a fairly simple problem, there is some combinatorial complexity here. So and you can leverage that if you know what you're doing. We've done some leveraging of that. One example is a cache efficiency improvement. Here's a case where we actually benched our code with an existing code that was in the public domain. It's not a particularly good code. It's secfold code. It had some mistakes in the energy calculation, but nevertheless, just to get an idea of like speed, right? We took this code from 2023, and we took their code and the secfold code and we just add our improvements to it. We called that secfold 2. We wouldn't recommend using it, but it gives us at least an idea about what the improvements can do for the speed up and the timing. And you basically, with these simple, additional steps, you can speed it up by about a factor of 2. So we're doing something that's useful, we think, for the literature on this. So these improvements were also used along with some other things to design the new GM fold code. So we use the energy function from Zucker and Stiegler. We use the step 2 from subgraph matching. We have a more robust multi-branch optimization than the Zucker-Stiegler idea, and I'm not going to go into too much details there. But anyway, it works pretty well. And the important thing here is not only are we getting results that compare well with the prior state of our M-fold, but it's in the public domain and you can parallelize this very trivially And, for example, we can compute the secondary structure of all 5,000 of these DNA sequences of interest. We can compute this in, a very short amount of time. I don't remember exactly how long, but we're talking about, a minute or two, kind of that time scale. Like it's sufficiently short that it's not at all of concern, for the research problem. So that's great. Um, so we have some comparisons between GM-fold and M-fold. We have some examples from the SecFold GitHub repository of sequences of interest, and you can see the lowest energies. Sometimes we're matching M-fold, sometimes we're close but not exact, and we're consistently beating the SecFold code, and we know why, because there are some errors in that. The errors are more at the level of maybe using the wrong energies for things. So, and shape-wise, um, here's our code in the third row or the third column. This is the M-fold. And some of these, like you can see the SecFold code and SecFold 2 are giving a sort of a larger multi-branch, middle region here than what we're getting over here. So there are a little bit of subtleties in how you do this, but nevertheless, here's a tough one. This is one where SecFold just completely messes up. SecFold 2 actually gets much closer to the right answer. Well, right, depends on what you call right. So let me just say that we can say that M-fold is the gold standard, but we still don't know if it's ground truth. So this is something to keep in mind that, you know, the model that we're using makes a bunch of assumptions. There's no expectation that it's 100% correct all of the time for what's actually happening in the chemistry. That's a difference between working on computational chemistry and some of the other machine learning problems that people like me work on in applied math. There's also this level of uncertainty in the underlying model itself. Okay, so yeah, so I mean, lots of examples. This is a biotin aptamer, and again, competing very well. Now there's an example. Here's an example where we don't perform as we'd like to. And there's a good reason for it. So this is theophylline. And the issue is that M-fold actually does include the possibility of a non-canonical pair, which we have decided we don't have that. So we could adapt our code to have this. It would add some more complexity to the code. And this is one where M-fold does allow for that, and it gets this shape rather than. So you have just one big circular region here, and here we have a couple of them because we actually are able to, we're able to include this non-canonical pair. Okay, and let me just say there's been a bunch of work in this. These slides are about a year or so old. Since these slides I've written these slides, there's many more people working on aptamer design, so it's becoming there is now more machine learning ideas and AI going into it. I think our work is a little bit special because we wanted something that was very interpretable, which means that we're working with more classical machine learning algorithms rather than black box AI methods. We think that's important. Okay, let's see, what else do I want to say? Right, okay, so the data set. So the data set that we've studied, we have some other data sets we're working with now, but the one that we've published is coming from it's a norepinephrine CellX screen. The work on the CellX was previously published. The lab protocols are in a number of different papers. Two different libraries were used, one with a 48 nucleotide randomized region and the other with a 58 nucleotide randomized region. The library design preferentially selects for aptamers with closed stem structures upon target binding and tertiary structure formation. This is a component that's salient in future biosensing applications, so there is a reason to do that. Data pre-processing. Okay, so now this is on our end. So the lab work is done. That's not my group's responsibility. But then they give us the data. The raw data is about 16,000 single-stranded DNA sequences. We remove the duplicates. We keep the sequences associated with the highest count. The count means how many times does that sequence appear in the next generation sequencing of the information. Remove all the sequences with less than 20 nucleotides or with an invalid nucleotide character. Remove the outlier sequences based on length. If they're really long or they're really short, we remove them too. We end up with 4,450 sequences, so 4,450 sequences with lengths varying from 33 to 83 nucleotides. Okay, so um, at this point in time, if we didn't have the machine learning component, what would happen is one would just pick maybe the highest count sequence and test that in the lab. So we want to show that there's a lot more that you can do with this. So what are we going to do? We're going to start looking at these sequences and we're going to take apart the secondary structure and we're going to look at the secondary structure as information that we can use to cluster, classify, however you want to think about it, these different DNA sequences. So there's a couple of things we look at here. One is taking the secondary structure and turning that into a function, and a unique function, a one-to-one correspondence. And this is using something called a Motzkin path. So the Motzkin path is measuring the cumulative sum of these bindings, and you're basically increasing by one or decreasing by one where you have the binding, and then you're flat where you don't have it. So if you just follow this thing around, you'll be able to map out the Motzkin path. So now we have a very simple, it's a discrete function. It takes on only integer values that are non-negative. And then we can basically use this as a substitute that's a one-to-one map to the secondary structure. But this information this doesn't include any information about the actual nucleotides and their order, just the shape. Now, for the nucleotides and their order, we can group them according to the faces. So we can look at different faces and we can treat this as like a topic modeling, like a dictionary, like a basically a document that's composed of words from a dictionary, and the dictionary are the faces. So we call this bag of faces. It's an analogy to the bag of words model from natural language processing. So this is basically like a natural language processing view of the DNA secondary structure. There's also a structural matrix that you can work with. The Motzkin path, though, is particularly nice because this gives us a high dimensional feature vector with, that's basically composed of integers. And that's a very nice kind of feature vector structure to work with for machine learning methods. Okay, let's look at the Motzkin path. So with the Motzkin path, what we can do is take those feature vectors and we can look at the t-SNE embedding. Not everyone here may know what a t-SNE embedding is. It's a t-distributed stochastic neighbor embedding. And it gives us a two-dimensional mapping of all of these nucleotides. So here they are in our two-dimensional space. There's different regions in the two-dimensional space. And then what we can do is we can color each node corresponds to a nucleotide. We can color them based on the count number. So the really bright ones have a high count, and the ones that are very dark have a low count, maybe just one. So the first thing we can do is we can do a very simple test of this hypothesis. We can take the top four high count sequences, X1, X2, X3, and X4, and we can look at this embedding and ask, is there something very close to the high count sequence using this embedding structure? Is there one that has just a count of one that's very close? And indeed, you can find this for all of them. And then we can look at what the structures look like. So for example, this high count aptamer here is almost identical to this low count one here. This has a count of one. It's not the same molecule, but it's pretty close. Here's another one, another pairing of a high count and a low count one. here's another pairing, and here's another pairing here. So this already gives us an additional 4 aptamers that we can try to test in the lab that might have a chance of being successful for for the things that we're looking for, mainly affinity and selectivity. So selectivity for norepinephrine, you'd want to see, could it bind to norepinephrine but not to similar neurotransmitters like dopamine and so forth. So that's the idea, serotonin, things like that. Okay. Then you can look at and topology and energy similarity neighborhoods for these aptamers, and you can look at things that show up within those neighborhoods. Um, you can use the bag of faces to compute energies of sequences with the same topology as the high count aptamers. So basically, we have a lot of machine learning tools at our disposal to be able to identify possible aptamers that would just not have come up using the prior methods that do not include the machine learning. We can use the topic modeling idea with non-negative matrix factorization to get topics, and then we can do spectral clustering based on the topics. And this gives us a different t-SNE visualization of the different clusters. So this is kind of an example of what we get with that viewpoint, that lens on this CellX screen. Where we are right now is we're doing some, not we, but Ann Andrews and her student, Saga Vallo, and also Ann's former student, Noelle Mitchell, are doing some lab experiments largely based on this Motzkin path similarity. And we're finding some really, really interesting stuff. And I'm not going to give away everything because we're currently working on a manuscript on that. So the takeaway here from the topic modeling analysis is even if the clusters are computed from high-dimensional descriptors, the two-dimensional embedding aggregates in specific regions of the plane. This suggests that the similarity of the high-dimensional descriptors is well represented in the low-dimensional space. Moreover, the geometric distribution of the two-dimensional representations points in the same cluster may aggregate in more than one region of Euclidean space. So the dimensionality reduction highlights additional hidden patterns between the sequences within the same cluster, which can potentially be exploited to segment the larger clusters further. We haven't actually explored these aspects yet in the lab, but the takeaway here is there's an awful lot you can do with fairly conventional machine learning methods to be able to really understand like the details of the structure of your different DNA aptamers that you're interested in. Okay, so bottom line is we've developed a new Python tool for the secondary structure of DNA aptamers. It allows for high throughput calculations of the secondary structure of thousands of aptamers. This is important if you want to use it to analyze all of the next generation sequencing coming out of your CellX screening process. It can identify the low count aptamers that are really close in structure to the high count aptamers. These would not have been easily identified with conventional methods. As always, further laboratory analysis of these aptamers is always needed for carefully measuring affinity and selectivity. So that's my talk, and thank you for coming to my talk.